Okay, so welcome back to this next video in which we are discussing acute gastric ulceration. Okay, right, so we're currently in the process of discussing the histological structure of the wall of the stomach. Okay, and this will be important in our understanding of the pathology of acute gastric ulceration. Okay, right, so at present we've just discussed the epithelial cell layer, which is sitting on this basement membrane. Now basically, underneath the epithelial cell layer, what you have is a layer of connective tissue known as the lamina propria, and basically the end of the lamina propria is marked by a layer of smooth muscle cells known as muscularis mucosae. And basically, it's easier to discuss muscularis mucosae first and then discuss lamina propria than it is to discuss lamina propria and then the muscularis mucosae, because the lamina propria just fills in all the gap between the basement membrane and the muscularis mucosae below. Okay, so this is a layer of smooth muscle cells here. Okay, and this is called the muscularis mucosae. Okay, so it's the muscle layer of the mucosal layer. Okay, so this is called muscularis mucosae. And basically I'll tell you what the mucosa is in a moment, and then all will make sense. Right, so now that we've discussed this layer of smooth muscle cells here, okay, and I'll just colour these in red to make them look convincing. Okay, on top of that, in between the basement membrane and the muscularis mucosae, all of this space in between is full of connective tissue. So all of this stuff that I'm colouring in yellow here, this is all connective tissue. So connective tissue everywhere, basically. And this connective tissue layer, this is what is meant by the lamina propria. Okay, so where can I put the label for lamina propria? The struggle. We'll try here. Okay, so this is lamina propria. So all of this connective tissue, and basically this connective tissue is going to contain blood vessels, and it's also going to contain some very important cell type, well, a very important cell type which regulates the secretion of hydrochloric acid by these parietal cells here. Okay, right, so it's a layer of connective tissue, and basically it fills in all of the gaps. So where you have these gastric pits with these gastric glands, clearly the lamina propria will be a very thin layer here, but where there aren't gastric pits, the lamina propria will be much thicker, basically. Okay, now underneath the muscularis mucosae, you then have another layer of connective tissue, and this is known as the submucosa. And now it really does become essential that we discuss what the mucosa is. But let me first just try and squeeze submucosa in there. There we go. Okay, so basically this term, mucosa, which I'll write here, mucosa, that means everything that we have discussed up to now, not including the submucosa. So it means the epithelial cell layer, the basement membrane, the lamina propria, and the muscularis mucosae, all of that together is then called the mucosa. So let me show this in vivid purple, something which will show up very well. So all of this, basically from here to here, this is the mucosa, basically. Okay, right, and now we can understand why this next layer that lies just underneath the mucosa is then called the submucosa. Okay, now what's in the submucosa? Well, you will have arterioles and venules. So here is an arteriole, and basically this arteriole will give off branches which then go up through the muscularis mucosae into the lamina propria and will supply blood to the cells within the lamina propria. Then also in the submucosa you have venules which will then drain the blood back. Okay, so the larger arterioles and venules will be within the submucosa, and they will give off smaller branches, or receive smaller branches in the case of the venules, from the lamina propria. And clearly, in between these, you'll have capillaries, which are supplying these epithelial cells and the other cells that are within the lamina propria. Okay, now what's underneath the submucosa? Well, underneath the submucosa, you start the muscularis layer of the stomach. Okay, so the proper muscle layer of the stomach. The muscularis mucosae was a pathetic little layer of smooth muscle cells. Now we're going to have the proper smooth muscle cell layers. Okay, so you have 3 smooth muscle cell layers. So I'll put these here. 1, 2, 3. Okay, now 1 is known as the oblique layer of smooth muscle cells. The next is known as the circular layer of smooth muscle cells, and the final is known as the longitudinal layer of smooth muscle cells. And to understand what this means, I need to show you a picture of the stomach again. So let's go back to the macroscopic anatomy of the stomach. Okay, so here we go. Here is our stomach, let's say. Okay, now basically the oblique muscle fibers will all be oriented in a certain direction, and basically the direction they will be oriented in is this sort of direction. Basically, this is the oblique direction. Okay, so all of the fibers that are in this layer will be oriented in this sort of a direction. Okay, so this is oblique. Now let's draw another little picture and put circular. Okay, so on this next one, I'll show you the orientation of the muscle fibers within the circular layer of smooth muscle cells. Okay, so basically in the circular smooth muscle cell layer, they will be oriented going around the stomach. So if you imagine the stomach is just a tube, and after all it is, then the axis is kind of, through here. Okay, so these will be going around the axis, if you like. So they'll be oriented in this sort of a direction here. Okay, sort of going around the stomach as though it's a tube. Okay, and that's the circular fibers. And then the next layer, the smooth muscle cells in the final layer, will be oriented in the longitudinal direction. So they will be moving along the axis of the stomach. So they'll be going like this. They'll be going towards the pylorus, basically. Okay, so the muscle fibers in these 3 different layers are all oriented in different directions, basically. Okay, so you have the oblique, the circular, and the longitudinal layer. Now, outside of the final layer of smooth muscle cells, i.e. outside of the longitudinal layer, what you then have is a final layer, which is a very thin layer of cells. Okay, so it's a single layer of cells, and this basically is the peritoneal covering of the stomach. Okay, so these are very flat epithelial cells. Okay, so this is the serosa, basically, and I'll colour this in a special colour so that it shows up. So I'll colour them in blue here. Okay, right. So it's often given this special name of mesothelium, okay, and that basically is to show, well, it's to demonstrate that it's derived from the mesoderm embryologically, basically, because that's where the peritoneal cavity, the pleural cavities, the pericardial cavities all came from. So the epithelium that lines the pleural, the peritoneal, and the pericardial cavities is all mesodermally derived. Okay, so that's why it's called the mesothelium. Right, okay, so that now concludes our discussion of the histology of the stomach. Okay, let's now look in a bit more detail at the secretion of hydrochloric acid by the parietal cells of the stomach. Okay, so I would just firstly like to discuss the structure of a parietal cell in a bit more detail, because they have a fancier structure than what is suggested by my picture here. So, okay, so I've not done them credit really in this picture here. I've shown them just looking like a purple columnar epithelial cell, but in fact their structure is far more exciting. Okay, so basically, if we draw a much bigger version of what we've drawn there, okay, So if we have, in turquoise here is the basement membrane. Basically, the structure of a parietal cell looks kind of like this. Okay? So they sort of move in like so. Okay, then they have a lump here where their nucleus is, and then they fold back like so. So this is the shape of a parietal cell, basically, and here is the parietal cell's nucleus. And basically, the columnar epithelial cells will sort of deform around this. So here's the neighboring columnar epithelial cell. Okay, and it will sort of make up this sort of bizarre shape here. Okay, and then we'll have another columnar epithelial cell, whoops, which is over here. Okay, and its nucleus will be down here as well. And I'll complete the basement membrane in turquoise down here. Okay, so we have a parietal cell sitting in the middle with these two columnar epithelial cells on its sides. Okay, now it's basically got this little channel which communicates with the lumen of the gastric gland, which will then go into the gastric pit, which will then go up into the lumen of the stomach. And this little sort of channel that it's got is known as a canaliculus. Okay? Right, so what's going to happen is this parietal/oxyntic cell is going to secrete hydrochloric acid into the canaliculus, and we want to discuss how it's going to do this. So what I'm going to do is zoom in on the basolateral membrane here and the apical membrane here, because this is what this is going to involve. Basically, we are going to be moving stuff from the basolateral side into the apical side. Okay, so let's have the basolateral membrane here and the apical membrane here. Okay, so the entire process of hydrochloric acid secretion is down to a special protein known as the proton potassium ATPase. Okay, and for short, the proton potassium ATPase is often just called the proton pump. Okay, so this is kind of its colloquial slang name. Okay, so let's colour the proton potassium ATPase in a special colour because it's, after all, the star of this show. Okay, so here we go, in orange. And basically what this protein does is it pumps two protons out of the cell. So remember, this is the cytoplasm in here, and this is the canaliculus over here, and this is the basolateral, well, this is extracellular fluid over here. Okay, right. So it's pumping two protons into the canaliculus. So this looks like a good start, basically. We were trying to pump hydrochloric acid in. Here's the first component of hydrochloric acid. Excellent. Now it wants to be electrically neutral, so it also needs to move two positive charges in, so it moves two potassium ions in, okay, and that means it's being electrically neutral, so this isn't disturbing anything electrically, basically, okay, and to do this, it requires the hydrolysis of ATP, so ATP will be hydrolyzed to ADP, adenosine diphosphate, and an inorganic phosphate. Right, and that will give the proton potassium ATPase the energy it needs to perform this movement. Okay, now what will this mean? Well, this is going to mean that the concentration of protons is going to go down within the cell. Okay, now there is an equilibrium that is occurring within the cytoplasm of the cell, and basically this is an equilibrium between water and carbon dioxide on one side, and then bicarbonate anions and protons on the other side. Okay, and let me just explain this equilibrium in a bit more detail. So basically, you can combine water and carbon dioxide together. So let me draw out the structures of water and carbon dioxide. So here's the structure of water, here's the structure of carbon dioxide. Basically, you can combine those two together to make carbonic acid. Okay, and we've already seen the structure of carbonic acid. Basically, you have a carbon double bound to an oxygen, and then you have two alcohol groups coming off. Okay, now carbonic acid is not a terribly stable acid. Okay, so it usually dissociates down to produce bicarbonate anions. Okay, and I should have drawn out the molecular this structure for this. Okay, so here's our bicarbonate anion again, and then a proton. Okay, so then you've got your proton here. Right, so it usually breaks down into this. So really what we've got is an equilibrium between water and carbon dioxide on one side and the bicarbonate anions and the protons on the other. And I want to stress this is not a one-way reaction. You can combine these together to make carbonic acid again, and then this will be returned back to carbon dioxide and water. So the stuff on this side can make this stuff, but this stuff can also make this stuff. So there's not a massive thermodynamic difference between the two sides. Okay, so the position of equilibrium isn't totally to one side, it's more in the middle. Okay, and basically the reaction going from one side to the other side is catalyzed by an enzyme called carbonic anhydrase. Okay, so let me try and fit this in here. Carbonic anhydrase. Okay, and basically what's going to happen when we suddenly decrease the concentration of protons? Well, the equilibrium is going to shift, because when you decrease the concentration of protons, you're going to decrease the rate of the backwards reaction. Now remember, what is the point of equilibrium? The point of equilibrium is when the rate of the forward reaction equals the rate of the backward reaction. That's what a dynamic equilibrium is. Basically, you reach an equilibrium when the number of water and carbon dioxide molecules that are combining together to give bicarbonate and protons in the cytoplasm equals the number of bicarbonate anions and protons which are combining back together to give water and carbon dioxide. Okay, so there's no net change to the concentrations on either side. But when you suddenly decrease the proton concentration, then the rate of the backwards reaction is going to decrease and the rate of the forwards reaction is going to be unaffected. So you're going to therefore start increasing the concentrations of the things on this side and decreasing the concentrations of the things on this side. Okay, and that will basically produce more protons which can then be pumped out via this proton potassium ATPase. So this is where we get this continual supply of protons from. Now of course you'll notice a problem here. Firstly, we're running out of water and carbon dioxide, but that's not a problem because respiration produces water and carbon dioxide. Those are exactly the two byproducts of respiration. Okay, so we don't mind that. But the other thing that's happening is that bicarbonate is going to go up and up and up and up within the cell. Okay, so basically what you then have on the basolateral side of the membrane, okay, here in turquoise, is a protein which can exchange bicarbonate, i.e. can throw bicarbonate out of the cell, and it will bring chloride anions into the cell. Okay, so both are negatively charged, so again this is an electrically neutral process, and I want to stress that all the way through this, that it's electrically neutral. Okay, so it's not disturbing the electrical potentials, the electrical potential differences across the membrane. Okay, right. So, you exchange the bicarbonate that's going up and up and up within the cytoplasm for chloride anions, which means the chloride anions are going to go up and up and up within the cell. Now, what else is going up and up and up within the cell? Well, also potassium here, because we're bringing potassium in and in and in. So, what now we're going to do is we're going to dump the potassium back onto the outside because we need it on the outside, because if we move it all inside, what's going to happen? We're going to run out of it on the outside, and then this isn't going to function anymore, because if it's got no potassium to bring back in, then it can't pump the protons out. So we need the potassium to be recycled back onto the apical surface. The problem is we can't just move it on its own, because that would, that would be moving two positive charges, and then we disturb electrical potentials. But what we can do is we can transport it with the chloride anions. Okay, so you'll have a potassium channel here, and the 2 potassium ions will move back out, and at the same time you'll have a chloride channel over here, and basically 2 chloride anions will also leave the cytoplasm at the same time as the potassiums. So again, we maintain electrically neutral movements, basically. Okay, so let's just colour in the chloride and the potassium channel. So we'll colour in the chloride channel in red here. Okay, and then we'll have the potassium channel in blue. Okay, so what overall then have we done? Well, the potassium hasn't moved at all, that's just recycling round and round and round, but we have secreted protons and chloride anions onto this apical surface. So we have put salt onto this apical surface, basically. Okay, now when you put a higher concentration of salt somewhere, what that will then lead to is water will follow the salt, basically. So we're going to get the movement of water from the basolateral side onto the apical side, and this will just occur by osmosis, basically. And then what we will have is hydrochloric acid on the apical surface of our parietal cell, which will go into the canaliculus, drain into the gastric gland, and then it will drain into the gastric pit and then onto the lumen of the stomach. Okay, so that's the secretion of gastric acid by these parietal cells. What we will move on to in the next video is discussing the regulation of the secretion of hydrochloric acid by these parietal cells.